Our goal in analyzing this data set is to generalize the results to the population that the sample was drawn from. In other words, if we see changes in brain activity in our sample, can we say that these changes will be likely seen in the population as well? To test this, we will run a group-level analysis, also known as a second-level analysis. In SPM, this means that we calculate the standard error and the mean for a contrast estimate, and then test whether the average estimate is statistically significant. Once all of the first-level analyses have finished, navigate to the flanker directory and type mkdir second-level _ inc-con. This will store our second-level results for the inc-con contrast. Then, from the SPM GUI, click on the button Specify Second Level. The default test that will be conducted is a one-sample t-test, and there are only two fields that need to be filled in: the output directory for the results and the contrast images that were created during each first-level analysis. First, double-click on the directory field and select the second-level inc-con folder you just created. For the scans field, navigate to sub-01's first-level directory and select the incongruent-congruent contrast image, con0001.nii. If you're unsure of what each contrast is, you can load the first-level results by using the Results button as we did previously. Contrast 1 is inc-con, contrast 2 is con-inc, and so on. Navigate into all of the other subjects' first-level directories and select the con0001.nii image for each subject. You can use the filter fields to do this more efficiently. When you have finished selecting the con0001 images for all 26 subjects, click the green Go button. Specifying the model only takes a second. When it finishes, you will need to estimate the model just as you did with the first-level analyses. From the SPM GUI, click the Estimate button. Select the spm.mat file from the second-level flanker directory you just created, and then click the green Go button. Like the first-level analyses, we can now view the results by clicking on the Results button from the SPM GUI. Select the spm.mat file from the second-level inc-con directory and click Done. You will see another contrast window, but with a slight difference. Whereas the first-level analyses had a design matrix that contained all of the regressors in the model, this design matrix looks like a white box. That indicates that there is only one regressor to test, namely the mean activation across all the individual contrast images that went into the model. Click Define New Contrast, call the contrast inc-con, and give it a contrast weight of 1. When you are finished, it should look like this. Click OK, and then click Done. You will be asked the same questions as before about masking, cluster thresholding values, and cluster extent. For this group analysis, select the following: no apply masking, no p-value adjustment, a threshold of 0.001, and extent threshold of 20. This will threshold the image to only show clusters that are composed of individual voxels, each passing a threshold of 0.001. In a later video, we will learn how to determine the exact cluster-defining threshold that gives us a false positive rate of 0.05. The threshold we're using now is just for convenience and shouldn't be used for publication. Afterwards, you should see output like this, showing a significant cluster in the dorsal ACC. Again, just like in first-level analyses, you can render these on a canonical template brain, found in the SPM12 canonical directory, to more clearly see where the activation lies. If you are only interested in where there are significant differences between the incongruent and congruent conditions, then the above steps are all that you need to do. As you will see in the next video on ROI analysis, however, it is useful to examine the activity in each condition separately to see what drives the effect of incongruent minus congruent. To prepare for that analysis, create two new second-level directories, one for the simple effect of each contrast, mkdir second-level _ incongruent, and also mkdir second level underscore congruent. Using the same procedure as before, we find that the incongruent contrasts are located in the O3 file for each subject, and the congruent contrasts are located in the O4 file. Starting with the incongruent contrast images, click on Specify Second Level, and then for the directory input, select the second level incongruent folder. Using a similar method as above, select the con0003 images for each subject. You will then do the same steps that we did for the inc-con contrast, loading all of the images and then specifying and estimating the model. Do the same procedure for the congruent condition as well. Now that you've finished running the group analysis, you are ready to do more specific tests using a method called region of interest or ROI analysis. All that and more in the next video. Although SPM comes with an extensive library for analyzing fMRI data, it doesn't have some of the tools you will need for more advanced methods, such as region of interest analyses. To fill this need, programmers and researchers have created SPM extensions, also known as toolboxes, which enable the user to do a wider range of analyses. For example, most fMRI researchers use some kind of atlas or partitioning of the brain into distinct functional or anatomical regions. fMRI data can then be extracted from a specified region and statistics run to determine whether there is a significant effect in that region. To create these anatomical regions of interest, we will download and install the WFU PicAtlas Toolbox, a popular atlas and region of interest generator. To download this toolbox, go to the website in the information box below. Select the atlas you want, and then click the download button. If you do not already have an account with the NITRC, you will need to create one before you can download the toolbox. Read all of this, or not, and then click Agree to begin the download. When the toolbox is finished downloading, unzip it, and then type the following code from the MATLAB terminal. Move file, downloads, WFU PicAtlas, and then your home directory, SPM12 Toolbox. This will move all of the needed folders into the SPM12 Toolbox folder, where they will be read each time you open SPM. Now open SPM, and then click on the Toolbox menu, and you should be able to open up the toolbox. MarsBar is another popular SPM toolbox, used primarily for ROI analysis. To download it, go to this website and click the download button. After the package has been downloaded, unzip it. Then, from the MATLAB terminal, navigate to the SPM12 Toolbox directory, and then we will create a directory called MarsBar, MarsBar. And then move the required files from the MarsBar download into the MarsBar folder. The next time you open SPM, you should see MarsBar as an option when you click the toolbox menu, just as we did with the previous toolbox. After you have installed both toolboxes, you are ready to do a region of interest analysis, which we turn to next. A region of interest, or ROI analysis, is a method of extracting data from a subset of voxels, also called a mask. One way to create a mask is to use an atlas, or a map that partitions the brain into anatomically distinct regions. Another technique is called the spherical ROI approach, in which a sphere of a given diameter is centered at a triplet of specified x, y, and z coordinates. These coordinates are often based on the peak activation of another study that uses the same or a similar experimental design to what you are using. In this tutorial, you will learn how to do both types of region of interest analysis. Let's begin by opening the WFU Pick Atlas toolbox you installed in the last video. The Human Atlas option will open an atlas of Brodmann areas, cortical parcellations based on tissue structure and cell organization. From previous studies of cognitive control, we would expect our study to show significant bold activity in the dorsal anterior cingulate or DACC region, roughly corresponding to Brodmann area 32. Click on the region Brodmann area 32 in the left menu, and then click Add in the middle panel near the top of the screen. The middle panel highlights in red the voxels that belong to the mask you created, which you can examine by scrolling through the slices. You may think that the mask is too thin to adequately capture your region of interest. In that case, you can enter a number in the dilate field and click either the 2D or 3D buttons to dilate the mask by the number of voxels you specify in either 2 dimensions or 3 dimensions. Enter a value of 1 and then click 3D and observe how the size and extent of the mask changes. Now click the Save Mask button near the bottom of the screen. Let's call this mask BA_32 and save it to the flanker directory that contains all of your subjects. Once you've created the mask, you can then extract each subject's contrast estimates from it. In this tutorial, we will extract the individual beta weights for incongruent and congruent separately, and then take the difference between the two. This allows us to determine how the incongruent and congruent beta weights combine to form the contrast estimate. First, click the Results button from the SPM GUI and load the spm.mat file from the folder Second Level Incongruent. Select the incongruent contrast, and for Apply Masking, select Image. Load the BA32 image you created using the Atlas toolbox, and choose Inclusive. This will restrict our analysis to the voxels inside the mask. Select No for ROI analysis and None for the p-value adjustment. For the uncorrected p-value threshold, set the value to 1 and the extent threshold to 0. This threshold will show all the voxels in the mask that we selected. Right-click in any of the glass-pane panels and select Go to Global Maximum, which highlights the current ROI that you've created. Then right-click again and select Extract Data, Whitened and Filtered Y, this cluster. This will output to the MATLAB terminal a list of each contrast estimate for each subject, for each voxel in the mask. To make this list of numbers more manageable and easier to interpret, type the following: inc equals mean y, 2. This returns a set of 26 numbers representing the contrast estimate for each subject averaged over all the voxels in the ROI. We will now do the same procedure for the congruent contrasts, loading the spm.mat file from the second level congruent directory and selecting the same options as before. When you have displayed the results within the BA32 ROI, extract the data, and then from the MATLAB terminal, type con equals mean y, 2. We now have a pair of 26 numbers, 1 pair per subject. We can enter this pair into a paired samples t-test using the following code. This will return 4 variables representing different parts of the hypothesis test. H is the result significant, 0 equals no, 1 equals yes. P is the p-value for the hypothesis, CI is the confidence interval, and stats displays additional statistics, including the t-statistic, degrees of freedom, and the standard deviation. You may have noticed that the results from the ROI analysis using the anatomical mask were not significant. This may be because the ACC mask we created covers a very large region. Although the ACC is labeled as a single anatomical region, we may be extracting data from several distinct functional areas. Consequently, this may not be the best ROI approach to take. Another technique is called the spherical ROI approach that we talked about earlier. This is considered an independent analysis since the ROI is defined based on a separate study. To create this ROI, we will need to use coordinates from another study. Let's randomly pick a paper, such as Yan et al. 2016. In the results section, we find that there is a conflict effect for a Stroop task, a distinct but related experimental design also intended to tap into cognitive control, with a peak t-statistic at MNI coordinates 0, 20, 44. To create this sphere, we will be using the MarsBar toolbox that we installed in the last chapter. From the SPM GUI, click on Toolbox and select MarsBar. To create a sphere, click on ROI definition, Build. From the Type of ROI drop-down menu, select Sphere and enter the coordinates 0, 20, 44. Enter a radius of 5, and for both the description of ROI and the label for ROI fields, enter DACC_sphere. Save the file to your flanker directory as DAC sphere ROI. Before we generate the ROI as a Nifty file, click on ROI definition and select View. Click on your newly created .mat file and make sure that the ROI is located in the region where it should be. If the sphere is in the right place, go back to the MarsBar ROI and select ROI definition and then select Export. In the Export ROIs drop-down menu, select Image, and then from the selection menu, click on the DACC sphere ROI.mat file. Leave the space for ROI image as the default and select the flanker directory as the output folder. Label the image as DACC sphere. We now have a mask that we can use for our ROI analysis, and we can use the same method as we did previously on extracting data from the anatomical mask. I'll leave the screencast running so that you can see how to do this. If you did everything correctly, you should get the following results, indicating that there is a significant effect. Okay, nice job. So just as before, we're at the very end, and it's a little less formal now. I'm not reading a script, but this is basically everything you would need to do to analyze a typical data set, typical fMRI data set from start to finish using SPM. Now, beyond this, there may be supplemental things like, I don't know, how to do bias analysis in SPM, how to do parametric modulation, how to maybe use other toolboxes. But for now, that's it. I'm glad you all stuck with this.